Okay, so today we're going to move on from talking about gymnosperms into talking about angiosperms. So this is our last main group of plants, our last division, Magnoophyta, and hopefully it will be spring after spring break and we'll start seeing some of these outside, maybe, cross our fingers. Okay. So Magnoophyta are the flowering plants. So we didn't see flowers on gymnosperms. We're going to start out with a very short video about the evolution of these plants before we get into some specifics.
Plants were undergoing their own evolutionary phase. This is Westwater Canyon in Utah. Ancient plant fossils of an entirely new variety have surfaced. Not a tree bird nor a gymnosperm, these plants had oval-shaped leaves. Their arrival would revolutionize the plant kingdom. In time, alter the ecology of the earth. Dr. Peter Crank, a biologist with Chicago's Field Museum of Natural History, has been sifting through layers of time, studying the origins of this great change. Rocks from the Utah desert are thoroughly washed in water, then gradually dissolved with weak acid to separate stone from plant life. Soon, a tiny flower petal shaped like a tulip emerges. The unlikely catalyst of major change in the Earth's economy. The angiosperm, or flowering plant. The ancestor of the modern-day magnolia may be the earliest flower on record. Inside the flower, a tiny gold bud coming out of the pupa stage. It's likely that relatives of this insect contributed to the revolution of flowering plants. Jurassic forests provided a sumptuous table for the goldbug and other new insect life. Pollen from flowering plants drifted through the forest like a soft rain. As the tiny insect moved from plant to plant, pollen stuck to its legs and was delivered to the flower's reproductive pistils, or seed. With the help of the goldbug, flowers spread across the landscape. Plants that received enough pollen developed a range of distinctive colors, shapes, and scents to attract more insects. In his outdoor laboratory, Cornell University biologist Dr. Carl Nicholas observes nature's age-old alliances at work. He believes that Earth's early plants and insects formed relationships for the same reason: mutual survival.
Insect pollination allows a plant to transport pollen from one individual to the next, using the brain of the insect to identify the same kind of plant as the donor of the pollen. So, for example, you can have angiosperms that are separated by many miles to flowering plants of the same species, separated by many miles, and the insect can transport pollen over that mileage. Plants can't move, but insects can. Gymnosperms tend to rely on wind for transport of pollen, so they can transport their pollen over a short, relatively shorter distance. So you can get widely scattered angiosperms that are still reproductively successful. If you widely scatter pine trees, they're not as reproductively successful.
The new flowering angiosperms had another advantage over other plants: the speed at which they reproduce. Gymnosperms, like cedars and pines, pollinated on the wind, and gained an evolutionary foothold years earlier. Once their pollen reached the pistil, it took a full 6 months to a year for the fertilization process to complete. The new flowering plants were dramatically more efficient. Once the pollen grains reach the stigma or on the pistil, they germinate, extending the pollen tube from the sperm to the ovary. Fertilization takes only 3 minutes after the infusion of pollen. Even the slowest of plants can fertilize within 24 hours.
During the Cretaceous, for the very first time, there was a partnership that was established between flowering plants and insects. Before that time, there was an antagonistic relationship. The insects were eating the plants. But with the evolution of flowering plants, a kind of hand and glove relationship began to evolve. Flowers started changing their reproductive structures to accommodate the insects that were visiting them. And insects were beginning to change their morphology, their shape, to accommodate the visiting of flowers.
Nectar produced by flowering plants is a powerful lure for their partners in evolution. New insects like bees and butterflies appear. New methods of pollination. Only one type of bee can help fertilize this ginger plant. Using its long tongue, it skillfully opens the petals of this flower. Pollen inside sticks to the bee's head and is later delivered to other flowers. The bee, in turn, is rewarded with nectar, off limits of other species. Through co-evolution, a specialized one-on-one relationship between flowering plant and bee is established. The cooperative effort produced an explosion of insects, with flora of virtually infinite texture and design. While dinosaurs may have dominated the ancient forests, insects and plants working together would survive the ages. Summer in Hokkaido, the northernmost island in Japan. Season when sweetbrier blossom blooms. Sweetbrier are relatives of the planet's early fruit-bearing plants. By responding quickly to evolutionary pressures, angiosperms won the cooperation of other species. Their first trial, attracting insects who would distribute their seed. Now, with the advent of fruit-bearing plants, came new partners in evolution. Mammals. Mammals disperse the fruit seeds to places they may never have reached otherwise. Both plants and animals benefited from this exchange. Ecologist Dr. Yumoto of Japan's Kobe University has has been conducting field research in Southeast Asia and Africa to study how plants and animals interact. Yamamoto has observed a symbiotic connection between flowering plants and mammals in tropical rainforests. Small fruit consumed by small animals like squirrels. Large fruit attracts monkeys and chimpanzees. Co-evolution to meet specific needs. As life becomes more complex, flowering plants and animals develop more exclusive relationships with one another. As large as a football, this giant fruit is a treat for the modern elephant. But if elephants can feast on angiosperms, why not other large creatures like dinosaurs?
Listen, totally agree more about. Dinosaurs have formed a symbiosis with plants by spreading the seeds and contributing to their reproductive process. And plants would certainly have made an adaptive effort to create large fruits to have their seeds dispersed more efficiently by them. But there is no evidence from existing fossils, either of plants or dinosaurs, that indicate there was ever a plant that benefited from such a relationship with dinosaurs. So, as angiosperms established an immediate relationship with mammals, was the close interaction emerged between angiosperms and insects, the pollen agent. There's a good possibility that dinosaurs were left out of the game and lost footing in the ecological system.
Okay, so that sets us up as our intro. And I will click through. Okay, so we are talking about angiosperms. Yes, an aside. Studying plant fossils is a real thing. If you want to become a plant paleontologist, I know a guy. I don't know if he knows me, but we can talk about it. I've met people out in the field studying plant fossils, and it's actually super cool. I highly recommend field biology. Okay, so we'll spend the rest of the course, honestly, talking about angiosperms. The last group we talked about the gymnosperms. There are maybe 1,000 gymnosperm species. But our guess for angiosperm species is something like 300,000, 500,000, lots and lots and lots of angiosperms. So if you have a stereotypical plant you think of in your head, you are probably thinking about an angiosperm. So just a reminder where we are in our phylogeny. Right, so if we're thinking about land plants, the bryophytes, the mosses broke off before plants developed vascular tissue. So then we had our ferns and fern allies, the pteridophytes, which pollinate by spores. Then we had the development of seeds. So last time we talked about the gymnosperms with their naked seeds. Now we're talking about the angiosperms. So in the angiosperms, we're going to see that the seeds are covered with a plant ovary. So they're protected. So when we say they're a covered seed plant, we can compare those structures. So up here on the right, we have a little bit of an example. So here B is our gymnosperm. So inside, we can see the megaspore. 3 is this structure called the nucellus. 1 is this integument. So this basically this shell. So that's what the seeds we saw in the gymnosperms would look like. We can see those same structures are present in the angiosperm, but now we also have this sort of capsule made up of an ovary wall that's going to be encasing our angiosperm seeds. If we think about typical fruits you might have in your kitchen, they are angiosperms. And so if you cut one open, grapes have been kind of modified through cultivation to not have visible seeds, but if you were to cut open a wild grape, you'd see it's full of seeds, so fertilized ovules, surrounded by this swollen ovary structure, which is the fruit. Same thing with an apple. False fruit does not mean an apple is not a fruit. What it's saying is that the part that you think of as the edible part, that's not actually the ovary. So the core, the fleshy part of the core, is actually the ovary for an apple. And you probably all seen the seeds inside of that core. Okay, so that's what we mean when we say that we're talking about covered seed plants. Okay. The reproductive structure for an angiosperm, is it suggesting music? Get rid of that. Okay. The reproductive structure for an angiosperm is a flower. So in gymnosperms, we talked about cones. Angiosperms, we're thinking about flowers. So we should become familiar with the different parts of a flower. So we can divide them up into male parts versus female parts. So over here on the right, what we see are the female parts labeled out. These three things together are also called the pistil or the carpel, or occasionally the gynoecium. So they're composed of a stigma that's going to receive pollen, a pollen tube within the style. So the style is this long, thin structure here. And then the ovary at the bottom that contains our ovules that are going to develop into eggs. On the left side, the stamen, also known as the Angiosperms, are the male parts of the plant. So these are made of little thin filaments with anthers on the end, and the anthers are going to be filled with pollen. So this is where we'll be developing pollen. Some other structures we want to be able to name when we talk about flowers. So our petals, here, the petals of a flower. So these can act as landing pads for pollinators, like these. My favorite flower actually is bat pollinated. I like it specifically because it's bat pollinated. So it blooms at night and the bats go up into it. I think that's so cool. Sepals, you would find below the petals. So those are like the little green parts standing out right where the flower is blooming. And at the base of the ovary, we have a receptacle. And then we have a little flower stem, that's our pedicel. So when we describe flowers, and we'll be looking at some flowers on Thursday as part of our lab, we want to know these parts. We're going to focus in now on the life cycle of an angiosperm. So we're going to talk first about the male parts, then we're going to talk about the female parts, fertilization, and pull it all together. So inside the Angiosperm, so inside that stamen, we do still have pollen. I talked about pollen with ferns, we talked about pollen with gymnosperms. The fact that it's an angiosperm doesn't mean it doesn't have pollen, it means the pollen is contained in a specific structure. We have these microspores originating from mother cells and what we call pollen sacs. So this is the anther of a lily, transected, so this is a cross section. So here we can see pollen developing in these pollen sacs. So here's where the filament would attach and try to support the nectar. So through meiosis, our microspores start to split apart, creating haploid cells. So as this microspore undergoes meiosis, becomes haploid, so we now have 4 cells. They then undergo mitosis, so that each pollen grain, so this is what's going to be dispersed by the pollinators, actually contains 2 different types of cell. So there's going to be a generative cell and a tube cell in our pollen grains. And actually, after we divide into generative and tube cells, we're going to divide again, and we're going to end up with 2 sperm cells coming from those generative cells. Meanwhile, in the gynoecium, so in the female parts of the plant, we're starting out with ovules here, right? Our ovules, embryo sac. Our megaspore is diploid, so that's our mother cell. It then divides into 4 megaspores, 3 of which die away. So 3 of them are aborted, and we have 1 surviving megaspore. And these 4 cells were created through meiosis. So they're not haploid. They then replicate, so they're undergoing mitosis until there are 8 nuclei, so basically 8 cells. And that's the point at which we kind of want to pause before we think about fertilization. So we're going to have in the center of our ovule, we're going to have 2 polar nuclei. So that's 2 of the 8. here we're going to have one egg. So that's the one that's going to get fertilized to eventually create our plant embryo. Then we have some other cells. So on either side of the egg, we have these synergids. And they're thought to kind of guide the pollen. And then we have 3 antipodal cells, which are going to nourish the egg cell. So during fertilization, we're going to have the pollen grain land on the stigma. So it's gotten there by being carried by some type of pollinator, potentially the wind. And this is a point at which I should note. So in angiosperms, when we're talking about fertilization, pollination, flowers can take their own pollen and self-fertilize. So if you've talked about Mendel in your other biology classes, he selfed his plants, right? So that he knew their genetics, knew they have the same genetics. But they can also fertilize other plants, right? So it can happen both ways, that a plant is reproducing with its own pollen, just doing it over again, or that we're mixing genetics with another plant. Okay. So the pollen coming into the stigma. It then extends its pollen tube that's growing out of it down the style. Okay, so we're going there. It has 2 sperm cells that are going to go in and get in contact with the egg. So this is how the embryo starts to develop. This creates an embryo sac. So we end up with 1 diploid zygote. And then the other sperm cell from the pollen is going to fertilize the polar nuclei. So those 2 cells in the center kind of merge together with the sperm as well, which ends up with a triploid structure, right? Not haploid, not diploid, triploid, because we have these 3 cells come together. And this is going to form the endosperm, which is a part of the seed that we're going to see that helps nourish the developing plant. So it's not going to become its own plant. It's going to become a structure within our seed. So here we see the life cycle outlined in full. So here on the right, we see that development of the pollen from the anther. So we have our microspore mother cell. We have that tetrad. We have the split into generative cell and tube cell. Then we have that pollen tube growing, that generative cell splitting into 2 sperm, going down into our ovule. On the ovary side, we have the ovary containing the ovule, creating that 1 main megaspore after meiosis, so splitting into 4 and then having 3 die away. And mitosis is happening, giving us 8 nuclei. The egg cell gets fertilized. The 2 polar nuclei also get fertilized together with the second sperm, becoming the endosperm. Okay. So we can see that in not quite real life, but helpful to see the movement. Double fertilization, which is what we're talking about, is special to angiosperms.
Reproductive process where there are 2 fertilization events. This double fertilization event occurs between the male reproductive organ, the male gametophyte, and the female reproductive organ, the female gametophyte. Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains 1 reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 megaspores degenerate, leaving only 1 surviving megaspore. The surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. This complete structure is known as the embryo sac. Within the established embryo sac, cell walls begin to form between most of the nuclei. 3 cells named antipodal cells form opposite the opening of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell,
This leaves two nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gametes, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen contains two main cells. A cell named the tube cell makes up the bulk of the pollen grain of the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce two hapless sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the two sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.
Okay, so that's our life cycle. Now we're going to touch back on the evolution of this group. Since we are talking about angiosperms now, this is kind of an intro lecture to the angiosperms, and we're only going to talk about two groups today, and then we'll continue talking about angiosperms in the future. But angiosperm diversity is pretty recent compared to other plants. So here we see a time scale versus basically how many different types of plants there are. So we can see that there is a diversification of conifers here in the Permian, Triassic, and Jurassic. We can see some of our other groups. We can see the ginkgos have been pretty solid, that same one ginkgo forever. And here we see that the angiosperms have really exploded rather recently. We can also look at this relative to other types of life. So here at the top, we see the angiosperms, so the in green, this top one. So we can see that around the same point, the angiosperms are diversifying. So as we're getting more different species of angiosperms in more different niches doing different things, we also have, especially within our insects, a similar explosion. So we start to see an expansion of pollinators, so bees and wasps as examples. We also have things that eat leaves expanding, so beetles, locusts, and bugs. You imagine eating pine needles, doesn't seem so nice as eating lettuce, right? So having larger leaves, more delicate, good for leaf-eating animals. And we also nectar eaters, right? They start to develop as now we have nectar in this new flower structure. So we can see that these two events are really lining up in an interesting kind of way. So this is a reminder of where we are in the tree, right? So we have our mosses, our ferns, our gymnosperms, and everything in green is angiosperms, these flowering plants. Now, angiosperm phylogeny gets rearranged a lot. In the past 20 years or so, there's an official angiosperm phylogeny group who kind of sets down the law, right, about what the official phylogeny is. So I believe their most recent version was in 2016, which is version 4. In your book, I think it's using the 2003 version, maybe. So this chart is a little different than the one you'll see in your book. But we can clue in on some of the groups. So I simplified it in that previous diagram, basically. What we're going to talk about today is a traditional grouping of angiosperms that we spoke about very briefly before when we were talking about the history of plant systematics. It doesn't exactly track according to modern phylogeny, but it's still something that's helpful to know about because it's still a kind of easy sort of term to use in the field if you're encountering a new plant. So the traditional division within angiosperms is mainly between the monocots, which you can see here on this tree, and then the dicots, which here have been renamed eudicots, so like true dicots. So here we see kind of everything else down here, more or less. So we can see that there are a lot of dicots also by looking at this. So as we go through following lectures, we'll talk about some of these groups within them. We're not going to be able to talk about each of 500,000 species, right? Even just talking about this whole list, it's pretty overwhelming. We'll be focusing in on things you can really see around here. So that brings us to angiosperm synapomorphy, so derived characteristics that we can see among the entire clade, among this entire group. So one of those is this double fertilization that we just looked at in their life cycle. So the fact that as we fertilize our egg using the pollen from the male structures, we actually have a second sperm cell that's also fertilizing the polar bodies, creating this new structure, the angiosperm, that's going to nourish our growing plant. Just the fact that we have this pistil structure, this female structure with the stigma, the style, and the ovary at the bottom, that's a unique characteristic of angiosperms. We don't see that in other groups. Same thing with the stamens. So those stamens and their pollen sacs, which come in pairs, that's something you'll see among all your angiosperms, but not in other groups. And there are also specialized cells and structures, especially within the phloem. So there's a companion cell structure in phloem, which we don't see in other groups. This is just a reminder picture that our xylem brings water up out of the roots, up into the plant, into the leaves, through the stem. And then the phloem is taking our sugars that we've created in the leaves back down to the rest of the plant. Okay, so we have specialized companion cells in the phloem. And the xylem also looks slightly different in angiosperms, has something called vessel elements. But we also saw that in one of our gymnosperms. So in our Nita, we saw the vessel elements as well. Thinking monocots versus dicots, this is something that's relatively easy to identify on site. One of the traditional way that they're defined is based on the seed. So when we're looking at the seed, we have our seed coat, which is going to protect the plant embryo. We have the endosperm, which is one of those structures that's created by the double fertilization attempt. So the one where we're not fertilizing the egg, the one where we're fertilizing the polar bodies. And then we have the cotyledons, which are embryonic leaves. So in a monocot, corn is an example. So think things like grasses. We have one embryonic leaf, a monocot is named for that. And then in dicots, so our bigger group, like a bean, for example, people like to use because you actually can kind of look at this in the kitchen. It's not that hard to pull a bean in half. There are two cotyledons, so two embryonic leaves. Corn is a grass. Corn is kind of a grass. So some things that we could potentially see without digging down and looking at a seed and splitting it in half. It's easy to split in half, but not all seeds are easy to split in half. So we can look at those reproductive structures themselves to give us an idea of this monocot versus dicot division. One quite easy thing to count is how many petals there are. So monocots tend to have their petals and their parts of the flower come in threes. So multiples of 3. There are obviously way more numbers of petals we could have than 3 or 4 or 5. But you may have like little clusters of 3s. Their pollen is also different. So you can actually identify plants based on pollen, which is something people do in research for all sorts of reasons, both to understand plants, but also like if you're an archaeologist trying to figure out what type of food a culture ate, you can look in pots and look for pollen grains and try and identify them that way. It's surprisingly kind of useful. So in a monocot, the pollen will have just like one little grain opening. But in a dicot, it's going to have the structure where we have 3 different grain openings. So we can see these kind of 3 sort of trefoil kind of structure, this 3-part. And the dicots, we're going to have more flower parts, so multiples of 4 or 5 in this dicot. Also takes us to the leaves. So when we come back from spring break, we're going to review all the labs, and we are going to focus in on our vegetative morphology review because we basically need to come back to that and sort of relearn some of those structures. But one of the things that we can add on when we look at it now is this monocot versus dicot division. So when we look at monocots, we're going to see that the veins run in parallel. So we're going to see parallel venation in our monocots. Versus in dicots, we're going to have branching patterns. So dicots are reticulate. That's another word for branching. And also cut apart monocot and dicot stems. So what we would be looking at if we do that, we'd be looking at the vascular bundles. So in this image, we have our vascular bundles, including both xylem and phloem. So our xylem is this darker green, and we have this phloem, this kind of olive drab color, and then another structure called the sclerenchyma. In a dicot, these are going to be in rings, all in a circle. But in a monocot, they're going to be sort of diffusely spread out, some sort of complex structure, or honestly, almost looking like a lattice structure. This should hopefully be triggering a thought in your mind when I say that the dicot stems have these rings of structure. Dicots have these rings. We've seen rings before in our lives, if not in class, right? So we think of trees as having rings, right? So when we're thinking about trees, and specifically hardwood trees, we are looking at dicots. So their secondary growth is forming additional rings with these vascular bundles in rings. So when we're talking about angiosperm wood, we call it hardwood. Gymnosperms do have wood as well. We just call it softwood, and we'll see a picture from a scanning electron microscope that can kind of show you why. So when we're thinking about woody plants, first we can think, is it a gymnosperm? If it's not, you're thinking about an angiosperm, specifically an angiosperm that is likely a dicot. So here we see the hardwood, so our angiosperm versus our softwood, so the gymnosperm. So you can see that the hardwood is much, much more dense, right? Everything's packed together. Whereas in our gymnosperm, we have a lot of pores here. So this physically makes it softer. We go down to the roots. We can see that in monocots, we usually have fibrous roots. So there's no like one main root, they're all kind of spreading out. Whereas in a dicot, we'll have a taproot. So one main larger root and then branches off of that. We could also slice through the root of a monocot and a dicot. So this arrangement, I just want to make sure that you don't confuse it with the stem, which is that way I wrote not the same as stem twice. So if you're looking at a dicot, its stem had rings, right? But if we're looking at the root, we're going to see that the xylem is in this kind of X structure or a star-shaped structure in the center. And monocot now, which before had these diffuse sort of vascular bundles in the stem, just all kind of spread out. Now we have the xylem in rings within the root. So that is the lecture portion of today. And now we're going to try my new experimental grading / recap method for our lab. No, it does have, some of you may or may not have heard of it. If you have your lab, pull it out. If you do not, I have the lab here. So what I want you to do is grab a lab key, colored pen, and your lab. Yeah. The faculty as a whole has a group going right now called ungrading. Obviously, we do have to assign you grades, but the point of it basically is that we should be focusing more on you mastering material than nitpicking point stuff, which sounds more fun for you and more fun for me, to be honest. So the way I'm going to try grading labs with this new method, we're going to reset up the lab. You're going to make your own corrections. So the goal for that is so that you can show me that you now understand it and you got it wrong the first time. And also write as many notes as you can, because then when you study from your lab, you'll know what you were looking at, right? Like knowing if you want to do question A does not actually help you if you're trying to study for a lab. Yes, whatever color that you have, I have my own. So long as it's a different color than you use it. Why not? If you got nothing wrong, then you're done and you can leave. Yeah, and I'm gonna set these up. So yeah, so basically what I want you to get the idea is a lab that is fully correct. So if you got it all right the first time and you understood lab, you have your time to yourself. If not, when you're setting up the lab, you're gonna correct them, and the goal is to like when you hand it back to me. Don't just write the answer that's on the key, right? Because that doesn't tell me you understand it now. That's just something that I can't do. Yeah. You're gonna write yourself a nice long explanation about why you got it wrong, why the right answer is right. There's not like a specific checklist. What I need you to do with your notes is just show me that you get it. So the more colored pen I see, if you got it wrong, the better. Oh, okay. Sorry. So I'm gonna put out the samples again, right? So the samples. Well, maybe you want to. Oh, I guess. After trying a couple times, I'll fold it. Like this.
Oh, so you just, yeah, we didn't think of that. Question. If you have a number where it's supposed to be an answer of a letter.
I think that you meant.
Did I miss it?
Like all the following answers supposed to go down one, maybe. Like from, are you on 14 or 13?
13.
Yeah, I think it's 13 something,
Can I
14,
remember
like
something?
14, 15.
Because this is two, then it's yes, then it's no.
Oh, I skipped one? Yeah, okay, sorry. Yes, 13 should be I, sorry, and then put them all down one. And I can display this on the screen and put the edits.
Oh, yeah, okay.